Computer's getting a little laggy. Be a good excuse to get a new one. Okay. So, another quote. I don't start every lecture with a quote here, but I thought this was kind of a cool quote. The brain is a monstrous, beautiful mess. Its billions of nerve cells called neurons lie in a tangled web that displays cognitive powers far exceeding that of any silicone machine we have yet to mimic it. That was back in 1989, where I think we're getting a little close to those silicone machines, at least in certain areas, certain types of tasks. They're getting pretty good. Kind of scary. So let's just go over the overview and start. We're going to go through some of this stuff pretty quickly. We've talked about some of these things already, so let's just kind of, but feel free to raise your hand, slow me down. Basic neuron, it's able to receive and generate signals and output through rapid changes in electrical potential, so that's very unique. It's got those four main components that Tori mentioned, the soma, the cell body, it's got dendrites, it's got axons, and it's got what we call presynaptic terminals, okay? Those are at the end of the axon there. With cell body, this is what makes up the cell body. The soma is you've got the cellular fluid by itself, just the fluid is the cytosol. Cytoplasm includes everything within that cell body except for the nucleus. Okay, so every organelle, all the cellular fluid, all that was considered part of your cytoplasm. Then you have the nucleus, which is where that genetic material is stored. Then you have that unique thing, that rough endoplasmic reticulum, the rough ER, which in neurons is called neutral substance, that's dotted with those ribosomes for protein synthesis that transcribe the little messenger RNA floating around that comes out of the nucleus, and then the ribosome transcribes it and builds the little neurotransmitters that are proteins typically to then be packaged. We have free ribosomes that are floating around within the soma that are making proteins to do things like rebuild the cell wall or make mitochondria and those kind of things that are for the internal use of the cell. They're not going to be for making neurotransmitters. So the free ribosomes are for producing proteins for cell use. The smooth endoplasmic reticulum, or the smooth ER, is for synthesis and transport of lipids. Okay, so, and then the Golgi apparatus. Sorting of proteins for different parts of cells and for cell packaging and packaging the neurotransmitters for making the little synaptic vesicles. So you have a very robust Golgi apparatus because it's got to do a lot of this packaging kind of thing. So. And then, of course, you got mitochondria because you got to produce energy because in order to keep that sodium-potassium pumps going, to maintain that resting membrane potential and to store those charges, and then when the action potential happens to reset it, it requires energy to keep charges apart from each other and maintain these electrical and concentration gradients. So when we talk about this and differences in cell membrane potentials, we got to think about the electrical side of things because these are charged particles, and the concentration part of things. Kind of a cool picture here that I've stole from some book. I can't remember which, but you can see here, see all this pink or whatever stuff in here? That's all that rough ER. This is all the neutral substance with the little ribosomes embedded in that neutral substance. Here's our mitochondria. Here's this big Golgi apparatus, and here's some vesicles that are being produced that have some neurotransmitter in them. And then you can see some of the ribosomes just floating, the polyribosomes that are floating within the cell that produce proteins for cell use. Here's the axon, and this is that region that Maria mentioned, the axon hillock. Right here is that place that joins the axon with the cell body, and then you'll see here's some myelin. We don't know. I don't know if this is a peripheral neuron or a central neuron, whether that's a Schwann cell or oligodendrocyte, but that would be the axon. That would be the myelin. And then when we get into this, we'll talk about an area right here near the axon hillock junction there between the axon hillock and the axon, an area called the initial segment. The initial segment, that is where there's a high concentration, typically of voltage-gated sodium channels that help initiate that action potential. And this is just a neat picture to show the dendrites. So you have all these dendrites that come off. They can be very numerous in nature. And then you have these little bumps, these dendritic spines that also come off that. All that does is enhance surface area and that's where you get all those billions and billions of connections, those whatever quadrillion connections or half a billion per cubic millimeter connections because of all those dendrites. But all the little dendritic spines that come off of those have the ability to connect or communicate or touch, get near another neuron. The cell membrane of a neuron is just like any other cell membrane. It's a lipid bilayer, and it has embedded proteins that are kind of embedded within that lipid bilayer. It's got, the lipid bilayer has that hydrophilic phosphate head, water-loving hydrophilic that tends to be face out or in towards the inside of the cell, the outside of the cell where the fluid is, right? Because it's hydrophilic, so it wants to be pointed towards where there's fluid. And then a hydrophobic tail that points in away from it on each side, so that creates that barrier because they want to be away from the water there. And that creates that barrier inside and outside. And we have embedded proteins that we call channels and protein pumps sometimes that help to move material and ions in and out of the cell membrane, in and out of the cell. This is just showing you that with all the little yellow things there are those hydrophilic heads, phosphate heads, and then the pointing in are the hydrophobic tails to create that barrier. And then this is just an example of a prototypical membrane protein that is typically made up of subunits, these polypeptide subunits. And what happens is you get, depending on the type of protein channel this is, some protein channels are just pores that they don't change shape, they just stay a certain diameter and allow certain sizes of molecules to pass through. Some are gated, and that's what we'll be focusing on today, is a lot of the gated ones that change based on whether a little neurotransmitter lands on it, or whether you stretch it, or whether you heat it up. But one or more of those polypeptide subunits it'll cause a conformational change in it that opens up that, at least temporarily, and then it usually closes after the stimulus is gone. So it allows this opening and closing of the channel based on the polypeptide subunits changing shape. The dendrites themselves, this kind of almost looks like a little squiggly tree or bush or something like that, and that's why it's called dendritic arborization. It's got a tree-like kind of look to it, or bush-like type of look to it. That's the dendritic arborization. Dendrites transmit information towards the soma. So action potentials come towards the cell body from the dendrites, or changes in charges come from that, from the dendrites. It's covered with thousands of synapses because those dendritic spines that I showed you earlier that has those, we saw in the little more closer up image. Danny mentioned the cytoskeleton, or some of the parts of the cytoskeleton that are somewhat unique to the neurons and give it its unique shape. It's basically the scaffolding of the cell, the skeleton of the cell, if you want to think of it. It's a good cytoskeleton is a good way to talk about that. So these are the three main components of that, the microtubules. So here you see the microtubules that kind of, using these tubulin molecules, these protein tubulin molecules that form this kind of tubular structure, this round tubular structure that's somewhat hollow in the middle. And it's instrumental in the transport of materials toward and away from the axon terminals. It acts kind of like a conveyor belt. So these tubulins are running all the way down your axons, and to move your synaptic vesicles down towards the axon terminals, down towards the presynaptic terminals, is they attach to this, and then they form these attachments, and then they release, and they kind of like move it along and facilitate, versus just these synaptic vessels happening to diffuse by chance down to the end. These guys are like little conveyor belts that help move the synaptic vesicles down. They don't move inside the tubulin, they're actually on the outside, and they just kind of form these connections and then form another connection that kind of pulls the synaptic vesicle along. And then also the same thing coming back. Once you've, squirted out some neurotransmitter in the synaptic cleft, some of that neurotransmitter actually doesn't get used and it gets reabsorbed, and also some of the components of the synaptic vesicle, the tubulin helps bring it back to get back and reused within the cell and then repackaged. Okay, so the tubulin is helpful for that. That's its primary role. Then we have microfilaments. These are just two strands of actin molecules that kind of twist like a Twizzler on each other, these little actin molecules. They're found in dendrites and axons, and they line the membrane kind of like a spider web, and they're involved in changing cell shape. And so you've got this lining of these actin molecules, and to make a dendrite push out or grow a little dendritic spine, you would just add more actin molecules onto the end of it, and it would push out that cell wall to form that. Or if you want to change direction of your dendrite to go over to this neuron over here, you could just build actin molecules that push that dendrite and grow that dendrite out to a certain direction. Or you can cleave off actin molecules on another side to shrink, on one area. So that just allows you to form and change the shape of your cell and adapt. And when we talk about neuroplasticity, that's what we're doing. We're helping to tell the cell, hey, grow more connections because we're using these neurons more often now, and so you need to grow a dendrite over here, so we call that axonal sprouting, when we want to grow an axon based on activity-dependent use and so forth. The actin molecules are involved with that. And then lastly, the neurofilaments right here. This is the one there is some microtubules and actin molecules in other cells, but the neurofilaments are very unique to neurons. So throughout the entire cell, the soma and the dendrites and the axons, you have these neurofilaments. They're analogous to kind of bones and ligaments of the body. They're polarized, and so you can see all of them in whatever that color is, a dark blue or purplish kind of color, but they're all spread out through here, these neurofilaments. The more neurofilaments you have in a cell, the larger the cell gets, because they want to stay away from each other. So if you keep packing more neurofilaments and they go, ooh, we don't want to be near each other, and they keep growing the cell, and then you take them out and it gets a little smaller. So it helps to dictate cell size because they want to stay a certain distance away from each other because of their electrical charge and polarity in those neurofilaments. Moving on to the axon. Each neuron has a single axon. Axons can branch. There are even times when the axon will branch back to its own cell body. So you'll have a single axon come off the cell body, but then you can have a branch come off the axon. The axon can divide at the end, but the axon can even branch back to its own cell body. There are certain types of circuits called reverberating circuits that kind of, once you get the circuit going, it tends to keep itself going. So even like certain reflex circuits and certain, like for walking, once you get walking, you just tend to keep walking until you have to tell yourself to stop. There's reverberating circuits that there's certain circuitry in that the axons and the cells and the neurons in that pathway, once you initiate it, they just want to keep themselves going until you tell them to stop. And that's, and they'll be self-stimulatory, and some of that's due to this kind of branching back to their own soma. The axon hillock we mentioned was the origin of the axon. Then we talked about the initial segment where at that transition point between the axon hillock and the axon itself, you get the greatest concentration of voltage-gated sodium channels. Axons typically transmit information away from the soma, and I said mostly here because I said there are select times when an axon will branch back to its own soma, but most of the time it's not doing. Most of the time it's carrying information away to go tell another neuron or an organ or a gland or whatever, an effector organ, what to do, or a muscle. Okay? Axons can be myelinated or unmyelinated. This is just showing you the axon here, but you can see there's branches coming off that axon. It's a single axon that comes off that. All the rest of those are dendrites, but it can have branches. This one's almost branching. It's not quite going back to the soma, but some do sometimes. But then you get down here and you see that it can really branch out to interact. So here you've got one big neuron interacting with a lot of little neurons, little second-order neurons. Okay. Then we're down to the presynaptic terminals of the axons. So now we're at the very end of the axon where those terminate. So we're down this region down right here where they're going to interact with the next either neuron, muscle, gland, some type of effector organ. Also called the axon terminal or a terminal bouton. So they're called the presynaptic terminal, sometimes called just the axon terminal or the terminal bouton. And that's where axons synapse another cell. Contains synaptic vesicles and a lot of mitochondria because there's a lot of energy that takes place in this process of releasing the synaptic vesicles, but also reuptaking and reusing what's left over as well. So there's lots of pumps. There's lots of chloride, calcium pumps down here because there's a little different process for that, which we'll talk about on Tuesday. Once the action potential gets there for releasing neurotransmitter, then reuptaking the neurotransmitter that's left over. General types of neurons. So there's unipolar neurons, which we don't have any unipolar neurons. These are only found in invertebrates. Simple single primary process. There's no dendrites or axons. It's just a extension that comes out. Bipolar neurons, we have very limited number of bipolar neurons, but they're used typically, you see them in special sensory functions. For example, retinal cells in the eye, olfactory receptors in the olfactory bulb, and then also the vestibular apparatus are using bipolar cells. It has two primary processes. It has an axonal root and a dendritic root, but it's only one axon and one dendrite coming out. Then we do have a thing called a pseudo-unipolar neuron, and where we see these in our bodies is just really in a peripheral sensory neuron. And so you have a single process that comes out of the cell body, which you would think is unipolar, but it immediately branches into two separate parts. It really branches into what we call kind of a peripheral axon and a central axon. And so that's the somatosensory pain and temperature proprioceptive neurons in our periphery, and that cell body for that pseudo-unipolar cell is sitting in the dorsal root ganglion. And then as soon as it comes out of the dorsal root ganglion, one branch of the axon goes back to the spinal cord and the other goes out to the periphery to whatever sensory receptor it's bringing information from. Okay, so that's a pseudo-unipolar cell. Then we have multipolar cells, which is what we see most of the time. One axon, but many, many dendrites. This is the most common neuron in vertebrates. Purkinje cells, this is the cell in your cerebellum, has up to 150,000 contacts with one cell. And you'll see, I'll show you a picture of that dendritic arborization of a Purkinje cell. That is pretty impressive. So here's the unipolar cell A, which we don't have. That's invertebrate neurons. Here's the bipolar cell that has dendrites and an axon, just a dendritic root and an axonal root. Okay, bipolar. This would be a bipolar retinal cell here. Here's our pseudo-unipolar cell. So this is the cell body here with a single extension or process coming off that divides into either a central axon and a peripheral axon. This is going to the skin or muscle. Sensory. These are sensory. This cell body is located in that little dorsal root ganglion just outside the spinal cord. Cell body for that. These are all examples of multipolar cells. So we've got multiple dendrites and a single axon coming out of it. Multiple dendrites, single axon. This is a motor neuron in the spinal cord. This is a pyramidal cell, the hippocampus. And then this is that Purkinje cell of the cerebellum. Check that out. So that's, when you look at the cerebellum again, remember how all the sulci and ridges, so it's got all that to increase the surface area and also the number of these cells you can put in that cortex. And then plus you've got these that are this many connections. So it just shows you how incredibly hard it is to do the processing to coordinate our body for the coordination, just how much impressive processing power that takes just by looking at the physical features of that. So good, looks like a good time for a break here. Let's come back in 10. record. All right, so we covered a little basics about the neuron itself, its structure a little bit, and what it looks like, physical properties. Now we're going to talk about the glial cells, which is actually more glial cells than neurons in your body, but they're not as relevant to us for PT clinical reasons, but sometimes they are. Functions of glial cells, they comprise half your total weight of your nervous system. They do not have action potentials per se. They do their charge can fluctuate a little bit, but they don't have a true action potential. They don't propagate signals down an axon or anything like that. They provide structural support, buffering, waste removal. They produce myelin, so that they are important for action potentials because they produce the myelin. Most tumors originate from glial cells. That's why you need to understand or know glial cells. Glioblastoma is the most common brain tumor, and those are glial cells, glioblastoma. Anybody take a guess why most of the tumors come from the glial cells, not from neuronal cells themselves? It's a feature of these cells, or they can divide and replicate much more easily and better. So cancer tumors are just a dysfunction in replication, where you get too much replication of cells, unchecked replication of cells. So types of glial cells, we can divide them into two broad categories: macroglial cells or microglial cells, or macroglia or microglia. But macroglial cells, some of the ones we've mentioned, are oligodendrocytes that form the myelin in the CNS. Oligodendrocytes are interesting because they will actually, the cell body will send these extensions out, then wrap around multiple axons nearby. Okay, Schwann cells, they form myelin around a single axon. They either fully envelop or they can partially envelop multiple axons. I'll show you a picture of what that looks like here in a second. So they can do either a single axon or partially envelop multiple axons. They don't fully envelop multiple axons, but picture in a second will show you that. Astrocytes. Okay, astrocytes they play a role in nutrition. They remove neurotransmitters, extra neurotransmitters that are nearby. They can pull and remove those. They also remove excess sodium afterwards. They help for the rapid repolarization. They can buffer potassium, and they also contribute to the blood-brain barrier. They form they have these little end feet that come off of them and wrap around the capillaries and create these little tight junctions where you've got this blood-brain barrier, which when you get to when you talk pharmacology about medications that are trying to get into the CNS, it's hard to get them in there because you've got this blood-brain barrier. So the molecules have to be extra small or have to be precursors. So once they get into the blood, then they're converted to another, chemical that turns into an agonist or antagonist to the neurotransmitter. But finding a way to get them past that blood-brain barrier is always the tricky thing. And that's formed by the astrocytes and these little end feet that come off of those. Microglial cells, these are primarily the cells, the scavengers that go and they had phagocytosis, ingest bacteria and dead neurons. They're the cleanup crew for the nervous system. Okay, a dysfunction of those has been implicated in Alzheimer's disease. There's all kinds of theories about Alzheimer's disease. Actually, Dr. Merriman was sharing these. He's writing a book chapter on lymphedema, and they've actually discovered recently, back in 2014-15, a lymphatic system in the nervous system, within the central nervous system, called the glymphatic system. And they're thinking that a dysfunction of the glymphatic system, its ability to remove waste from the nervous system, is one of the reasons why you get this buildup of tau proteins with Alzheimer's disease. So that's another thing that they're thinking, is that there may be a dysfunction in microglial cells, and they're eating up neurons and damaging neurons when they shouldn't be, and then also you're getting a backup of waste and products and protein deposits because of maybe some dysfunction in the glymphatic system. So kind of interesting. This is just showing you this dendrocyte, the cell body here, but you can see it has projections that go and it surrounds multiple axons. And it doesn't just typically surround these things with one layer. They wrap around like a cinnamon roll, almost, multiple times around those axons. Same thing with a Schwann cell. So here's the Schwann cell, and this is the axon. Here's the cell body, and this is the actual cell. It does this whole wrapping around the axon multiple times, okay? And that is considered, when we say a peripheral neuron is myelinated, it has to have that Schwann cell wrapping around it multiple times. You can have Schwann cells that envelop multiple axons nearby, but you see there's still a little gap there. They don't fully wrap around. Those are still considered an unmyelinated axon. Even though there's myelin, a Schwann cell that is partially enveloping it gives it some support and it keeps those axons from moving around or interacting with each other, but it doesn't do a good enough job to really help action potentials propagate down them. You actually have to wrap around. You got to form a good insular layer by wrapping around it multiple times than just partially around it. So that would still be considered an unmyelinated neuron. Here's that astrocyte, astro, star-like, and it looks kind of like with all these projections, kind of like boom, like an star, and it's got projections. So here's these end feet that can project, and the end feet can kind of stabilize neurons, but then some of the end feet project and form these tight junctions between them and wrap around the capillaries and form that blood-brain barrier, is what they do. And so they're the glue. Again, they're gluing everything together, so you just don't have neurons sliding around against each other, and it provides a little separation or buffer between neurons. So you're just getting more of your connections in your neurons between the actual intended dendrites and axons and so forth. That's all we'll say about glial cells right now. So let's get down to business as far as the actual transmission of information or changes in membrane potentials and these action potentials that are the cardinal function of the neuron. So, in order to have an action potential, you actually have to start with a difference in charges between sides. You got to have this potential difference so that you can have a flip-flop of charges to send a signal down the axon. So, let's talk about how do you establish the resting membrane potential in the first place. So, nearly all cells do have some difference in electrical potential. However, nerve and muscle cells are the ones that are mainly the ones that are excitable, that they can actually change potential, flip-flop that charge from the inside to the outside. Membrane potentials are established by different concentrations of ions on either side of the cell membrane. Okay, so it's charged particles, differences in concentrations in those. The primary ones we're going to talk about for establishing the membrane potential are going to be potassium and sodium. They by far outweigh all the other ones in how they influence the resting membrane potential. There are others. There's chloride ions, there's calcium ions, and sometimes those chloride and calcium ions are more important in action potentials in, like, say, cardiac muscles, or what's happening at the presynaptic terminal for helping synaptic vesicles move towards the presynaptic terminal and release their neurotransmitters. So just understand, we'll get to some of those, especially when we talk about synaptic transmission next time, but we're going to focus mainly today on just what's happening with potassium and sodium. And then we mentioned before, and Danny mentioned about the negative, or was it Kyle? I think the negatively charged anions in the that are stuck inside the cell, these phosphate groups that are stuck inside the cell that also help to keep some negativity on the inside of the cell. So we're talking about, when we talk about resting membrane potential, it's the inside of the cell relative to the outside of the cell. So here's this nice little microelectrode with a little probe determining what the voltage is on the inside of the cell, and here's a ground that's telling us what the voltage is on the outside and comparing those voltages. And the resting membrane potential is typically a minus 65 millivolts, which means the inside of the cell is 65 millivolts more negative, on the inside of the cell compared to the outside of the cell. Okay. And so, and these charges, these negative and positive charges, since they actually attract each other, there's still a little bit of an attraction across this cell membrane. They really kind of line up right along the cell membrane. Those negative charges and positive charges all want to get close to each other because they're attracted to each other actually, but they can't interact because you've got a cell membrane there. So just understand that even though you have inside your cell, you have these positive and negative charged ions, they tend to line up right along the cell membrane so that you can get that quick change of charge when the action potential actually occurs. They're not evenly distributed throughout the cell. They tend to line up along there because they're kind of attracted to each other. So inside of the cell, this is in an axon here. They're cross-sectioning through this axon of this neuron. So this is inside right here. So this is inside concentrations. And then this is the outside area here. So this is outside. So for potassium, on the inside of the cell, 100 millimoles concentration. Oh, sorry. This is the ratio outside to inside right here, I should say. This is outside over here, the outside concentration. Potassium is 5 millimoles on the outside, 100 on the inside, a 1 to 20 ratio. Sodium is 150 millimoles on the outside and 15 millimoles on the inside, so a 10 to 1 concentration outside to inside. Okay, and the way that it gets that way is because of the sodium-potassium pump. So the sodium-potassium pump, it's pumping potassium into the inside of the cell and pumping sodium out of the cell. And that's the only way you can establish that gradient there, okay, is through, requires energy and work to move things against their concentration gradients and or electrical gradients, okay, to move them against which way they normally want to travel. So the ionic basis, so membrane potential is that voltage difference across the membrane. The resting potential for a typical neuron can vary between minus 65 millivolts up to minus 90 millivolts, depending on the size of the neuron, where it's located. It varies between that. Negative, always referring to the inside compared to the outside. Okay? Each ion contributes to the resting membrane potential differently, depending on what's called its electrical chemical potential, okay, or its Nernst potential, and its individual membrane permeability at any given moment, okay? And we'll go through all this and kind of tease that out a little bit, help you understand that. Remember, these are charged particles, so there's an electrical gradient. Charged particles either want to, if their opposite charges come together, or if they're negative charges, if they're the same charge, be away from each other. And then also the concentration gradient. So you have to always factor those two things into when you're talking any ion or charged particles. It's electrical gradient that's going to push it or pull it, and it's chemical gradient, concentration gradient, that's going to push it or pull it in a different direction. We talked about what maintains that. It's that active transport of sodium and potassium. Each time that little sodium-potassium pump works, it's pumping 3 sodiums out and 2 potassiums into the cell. Okay, and that's what's keeping sodium out and putting potassium in all the time. It's always running. That sodium-potassium pump's always running, because we'll find out there's a reason it has to keep running, because the potassium channels are leaky, and so there's always some leak of potassium, and we'll get to that in a second here. So there's also some passive diffusion. That's what I'm talking about, is there's some passive diffusion, and sodium and potassium channels to non-gated ion channels. So there's some just protein channels that are sitting in there that are non-gated. They're leak channels, we call it. And there's a lot more leak channels for potassium than there is for sodium. So there tends to be some leakiness for potassium. And then the negatively charged anions, these proteins and organic phosphates and sulfates that tend to be more negatively charged, they're trapped inside the cell and too large to move through the cell membrane. Things like water and oxygen, those things just tend to, even though like water has some polarity to it, it's a small enough molecule, and it can move through that cell membrane. Same thing with like oxygen, can just move through the cell membrane. They don't need channels to do that. But most other things require some channel to do that.
How can water move through if the tails are hydrophobic?
They are, but they're not super hydrophobic and they move fast enough that they can kind of punch through? Yeah, that's a good question. So they're not quite hydrophobic enough. So, yeah. All right. So sometimes it's easy to let's look at the two main ions and how they influence the resting membrane potential, and look at them individually first. Okay. So we're going to set up some artificial conditions here to simulate the situation. So let's start on the left, and let's just look at potassium. So we're going to start with a high concentration of potassium on the inside. That's what our potassium pump set this up. But right now we're just showing this. This is a cell membrane. This potassium pump is pumped more potassium into the inside of the cell, so there's a higher concentration of potassium on the inside. And right now there's no protein channels and no way for that potassium to leak back and forth. And so you have a smaller amount of potassium here. These are currently being balanced by these negative anions. So right now there's no difference in charge between the inside and outside of the cell. However, there is concentration gradient differences, right? There's concentration gradient difference. We're just going to focus on potassium. Don't worry about these guys right now. That potassium, if we did stick a channel in there for potassium, it would want to move out of that cell just purely because of a concentration gradient. Even though there's no electrical gradient right now, because these are balancing each other, there's equal positives and negatives on the outside. The only thing that would influence it right now would be when you first put that protein channel in there to leak it, allow it to go through, it would be just the concentration gradient, right? So we do that. We put a potassium channel in there that's just open. Potassium starts to leak or move down its concentration gradient, but very quickly, because it is a charged particle, as it's leaking and it's moving down its concentration gradient, it's bringing positive charges to the outside of the cell, which are going to immediately line up right against the cell membrane, and it's leaving the inside of the cell more negative because no longer is it balancing out that negative anions there. So you're getting positive charges are leaving the inside of the cell, but nothing else is. So that means the concentration gradient is currently strong enough to overcome the electrical gradient, right? Because you're moving these potassium ions are moving now very quickly. Once you start some leave, it's getting more negative on the inside and more positive on the outside. So potassium eventually, that chemical gradient is going to be superseded by the electrical forces. They're going to eventually then kind of balance it all out, because eventually you can only move positive charges towards that positive environment. You have to have a strong enough chemical gradient to overcome that electrical gradient initially, and then eventually the chemical and electrical gradients balance out. So at a certain point, once enough potassiums move over here, the electrical and chemical gradients balance out. So you can no longer, the chemical gradient is no longer strong enough to overcome the electrical gradient that's going to push potassium, to keep potassium from going out into that positive outside of the cell, and also the negative wanting to keep it in and pull it back because of that electrical charge, okay? In a neuron, typically that balancing point, that electrical chemical balancing point, when you set that up, is somewhere around that, we could say minus 65 to 90 millivolts. So when the inside of the cell becomes negative by between minus 65 to minus 90 millivolts, potassium no longer wants to move across that cell membrane. Does that make sense? The electrical chemical gradient has kind of been neutralized at that point. Then we have basically the exact opposite for sodium. So sodium, the sodium-potassium pump has pumped a bunch of sodium to the outside of the cell. So it has a strong chemical gradient that wants to bring it in right now. Right now we've got a balance between positive and negative charges, so right now there's no charge influencing it. But as soon as we open a channel here, some positive sodium start flowing in. So now the inside gets to be more positive. But you've got still here a strong enough chemical gradient that keeps drawing sodium in, even though it's getting more positive in here, more negative here. But eventually, once enough sodium moves in, You've got enough positive charges that prevent more positive charges going in, and enough negative over here that want to retain whatever, kind of retract sodium, and then they balance out, and you stop getting flow through that open little channel. For sodium, that is about a positive 62 millivolts where that occurs, that equilibrium potential, that Nernst potential, where it reaches that electrical chemical equilibrium, where you don't have any more movement of the ion across the cell membrane. So if you'll notice, so the equilibrium potential or Nernst potential for potassium is that minus 65 to minus 90 millivolts, where the equilibrium potential for sodium is plus 62 millivolts. We know that the resting membrane potentials for neurons are around that minus 65 to minus, they're the same as potassium. Why is the resting membrane potential closer to potassium's resting membrane potential than sodium's? I kind of mentioned it briefly earlier. Does anybody have? Yeah, because there's a lot more of these little leaky channels for potassium. So this is always occurring in the background of a neuron. Is you're pumping potassium in, but it's also leaking out pretty quickly. So you've got this sodium. So that's why you have this resting membrane potential that's closer to potassium's equilibrium potential by itself. Because sodium there's a few sodium leak channels, but not enough. It's inconsequential, for that. So they provide a little bit of positive leaking in, but not enough to counteract the leaking out of potassium all the time. So that's why the resting membrane potential is closer to potassium's equilibrium potential, is that potassium just tends to be leaking out all the time, kind of similar in this fashion. But that's why those sodium-potassium pumps just keep cranking, because this is always happening. They just got to keep that cranking going on of like, oh, potassium's leaking out, we got to put it back in. It's leaking out again, we got to put it back in to maintain it, at this level, and keep that negative, 65, 90 millivolt difference between those, okay? Just to freak you out, so we said sodium and potassium are important for understanding that. There's lots of things that dictate or contribute to the resting membrane potential. So this is the Goldman-Hodgkin-Katz equation. This is the voltage difference, the difference between the inside and outside, and it's based on a lot of things. So it's based on the concentration of the potassium on the outside and inside, sodium, and they also include chloride here. Other things actually, you could put calcium in there too as well. But some of them get almost inconsequential in how they affect, because if they're not very permeable at all, then they don't really affect the resting membrane potential very much. And so you've got concentrations of potassium, the ratios of concentrations between the potassium and sodium and chloride, the inside and outside. Then the P is the permeability of each of those, how permeable, how many leak channels, and how fast those are moving. And then you have things like the universal gas constant, temperature. How fast molecules will move is also dependent on the temperature of the system and how fast those things move. Faraday's constant. So you've got all these things that influence, and you multiply that out, and that gives you the true equilibrium or resting potential, I should say, resting potential of the membrane at any given moment. Based on, and that can change based on the temperature and other things happening around the environment of the cell and the moment-to-moment permeability of those things. So now we got to talk about, now we've established the resting membrane potential because of that sodium-potassium pump and the leaky potassium. Those are the two main things that get the resting membrane potential, is that sodium-potassium pump and the leaky potassium. But then we got to talk about how we get changes in that resting, those temporary brief changes in resting membrane potential. When a cell is depolarized, it becomes less negative inside or more positive inside. So a depolarization of a cell is it's becoming less polarized, so it's getting more close to zero. So depolarization, which is getting more positive or less negative. So that's depolarization. And at a certain point, it actually shoots above zero and gets even positive on the inside of the cell very temporarily. Of the neuron. When a cell is hyperpolarized, there is a brief period of time where your neuron actually gets more negative. Okay, so it's called hyperpolarization, which is when we're resetting the action potential as a temporary period of time when so much is rushing out of potassium and so forth that you get a little bit you actually get a little extra negativity briefly below the resting membrane potential. Okay, that's hyperpolarization. All right, so we got to talk about the establishment of the membrane potentials and then how we initiate action potentials and the requirement, and it requires movement of ions across the cell membrane. So back to real basic kind of chemistry and physics here. Ions move, and they can do it a couple different ways. One is through what's called diffusion, okay, or chemicals. It doesn't have to be ions. It can be any molecule can move because of what's called diffusion. We have two different types of diffusion, simple diffusion and facilitated diffusion that we're going to talk about with neurons. The other one is active transport, okay? So simple diffusion. Simple diffusion, where you get molecules moving, is based on either a concentration and or electrical gradient. If it's a charged particle, it's going to be affected by electrical gradients as well. This can occur directly through the lipid bilayer for certain molecules, oxygen, nitrogen, carbon dioxide, water molecules. Those can, move through, some of them better than others. Some of them don't. Water molecules move some, but they don't move through as easily as like an oxygen molecule without any protein channels or anything like that, because they're small enough and they're not charged enough to be affected too much by that. Then we have ones that have to go through protein channels, okay, and those are going to be most all ions are going to need to use a protein channel to get through. And lipid insoluble substances are going to also, most of them have to use some type of ion channel. And the types of channels we're going to talk about, these protein channels, are either non-gated, means they're just open and they just move through. They may be specific to a certain size or diameter or type of molecule moving through it, but they're not gated. They don't open and close. They just stay the same all the time. And that's where there's a lot of those for potassium. In a typical neuron, there's a lot of non-gated, where potassium can leak through. Then we have modality gated. Okay, so those are ones that are like temperature, stretch, vibration, whatever, that can cause protein channels to change shape enough to allow whatever, oftentimes sodium, to start moving through that. Then we have ligand-gated channels, which are channels that require some kind of a chemical bond that lands on a receptor on that protein channel that then causes the change to occur temporarily. As long as that little ligand or that neurotransmitter is bound to that, it'll stay open. Then once it releases, it'll close. Okay. And voltage gate is the other one. The other big class is voltage. So voltage gated channels respond to a change in voltage nearby, a certain threshold value. When a voltage changes close by to these channels, they recognize that change in voltage. Once it hits a certain value, they go, oop, it's time for us to open. And then once that voltage changes again, they close. Okay? Or they have some of these have a timing mechanism, so you don't need anything to close them. You initiate it, and they stay open for a certain amount of time, and then they close automatically. Okay? Usually we're talking milliseconds here when these kind of things are happening. So here's just showing you some of the examples. So here's a ligand-gated, so this would be maybe a neurotransmitter binding to a specific receptor that once it binds on it, it causes a conformational change in one of these polypeptide subunits that opens that up. And this may then, once this thing releases, then that goes back to its closed state. Okay. Here's a voltage-gated, so here's the resting membrane potential. You can see here's normally we have it's negative on the inside of the cell, so this is down here is the inside and that's the outside. But now we've got a change in membrane potential. We've got an action potential going down the axon here, and we've got a flip-flop of charges. Now the inside is more positive and the outside is more negative, and that causes that conformational change in some polypeptide subunits to open up and allow temporarily ions to move through there, whatever that's specific to that protein channel. And then here's one, you've got these little these could be some little microfilaments here that are attaching the actual protein channel to the cell membrane here, and so this could be on or a receptor out in your skin, and you push on that, a pressure sensor, a Merkel's disc or a Pacinian corpuscle that has some stretch, modality gated stretch sodium channels, and when you push on that, it opens up enough that the action potential starts down here in my finger to send a sensory signal up to my spinal cord saying, hey, I'm pushing this hard, or I've touched this object. That's modality gated. The other type of way that molecules get through is what's called facilitated diffusion. And these are facilitated type diffusion where you got to get some conformational change in a carrier protein that allows ions to cross the membrane. But the rate is limited by how fast that protein can change. You know, how fast and big or whatever that protein can change or stay open. The rate with which that occurs is mediated by that protein. Okay? Facilitated diffusion still requires a concentration and or electrical chemical gradient. This is not an active process. This is not active transport. This is still has to move down some kind of a chemical or electrical or combination gradient to move across. Okay? So facilitated diffusion. There's a specific type of facilitated diffusion called carrier-mediated diffusion, where certain molecules like glucose and most amino acids use a process of co-transport. They use another molecule's concentration or electrical gradient to help them get across the cell membrane and move against their concentration gradient by using somebody else's concentration gradient. So you may be trying to move glucose into a cell, and you already may have a higher concentration of glucose in the inside of the cell and the outside of the cell, but glucose may use co-transport with a sodium ion to move into the cell at the same time and use its concentration gradient to move glucose against its concentration gradient. So here's a very rough idea of that. So here's a carrier protein here that's causing this facilitated, carrier-mediated diffusion. So you've got receptors for both glucose and sodium here. You get a sodium ion binding here and a glucose, and then sodium wants to move into the cell, right? Because there's a much higher concentration on the outside. It's negative. So sodium wants to move in. So sodium will help then to bring glucose, even if the glucose concentration is already higher in here, which it wouldn't be able to move by itself against that concentration gradient. So it uses the concentration and electrical gradient of sodium to help pull it in with it. But it's limited by how fast that process. You can only move glucose so fast in the cell based on that little protein resetting itself, opening back up to the outside, bringing the next one in. So there's a limited rate there. So the factors that affect this rate of diffusion would be the permeability of the membrane, the differences in concentration between the outside of the cell, that concentration gradient, and then the electrical potential between the difference between the outside. The stronger the electrical gradient, the stronger the concentration gradient, the faster, and these things are going to want to move, given that they have the permeability. The rate limiting thing is the permeability. If there's no channels for them to get through, they can't do anything. So, and that's what drives the action potential, is that rapid change in permeability in membranes as it occurs, and how it occurs sequentially down an axon that allows you to actually directionally propagate this change in polarity down in a directional so you can actually get information going in a certain direction. Then there's active transport. So what we talked about, simple diffusion, facilitated diffusion, don't require energy to do that. They just move using concentration gradients of some time or electrical gradients. Active transport, you're moving things against those gradients, okay? And so that requires ATP, requires energy to do that, to move things against those. So they use a carrier protein, we typically call the sodium-potassium pump. There's also a calcium pump that does a similar thing for calcium. And so here you see this sodium-potassium pump, when it's open to the inside of the cell, it will take 3 sodiums from the inside while it's dumping out 2 potassiums to the inside. And then when it opens up to the outside of the cell, it'll release those 3 sodiums to the outside of the cell and bind a couple potassiums, so that the next time it changes, it'll do just the opposite. So just kind of this pumping action of pumping out 3 sodiums and bringing in 2 potassiums for every time it does that cycle. Okay? And that's why you end up ultimately with more sodium on the outside of the cell, more potassium on the inside, but then potassium's in the background is always doing this little leak thing that makes that resting membrane potential closer to potassium's equilibrium potential. Let's talk about this, a little subtle difference here. I don't want you to get confused between what we call local potentials and action potentials. Okay? So a local potential is a local graded potential that are smaller changes in these membrane potentials in a certain area of a neuron that when you add them up, if you get enough local potentials, if you get enough, we call it either temporal or spatial or combination of temporal or spatial summation of these local potentials, that could lead to an action potential if you get to the threshold value, the change in membrane potential that gets to this threshold value. Then that causes a bunch of voltage-gated sodium channels to finally open, which causes this rapid change in polarity and causes the action potential. Sometimes they're referred to or classified as a receptor or a synaptic potential is another term. So local potential, receptor potential, or synaptic potential. And it'll make sense here in a minute when you see the pictures. And then action potentials are the brief, large, all or none responses. Either you have one or you don't. And when you have one, it's going to happen. Okay? If you don't, it's not, you know, so this is the action potential, the brief, all or none change in membrane potential that can be then propagated directionally down an axon to communicate with other neurons. And it requires you reaching a certain threshold value change in potential in the neuron before that will occur. Because you have to add up these local potentials to usually get to that threshold value. So here is an example of the local potentials, and then here's an example of a temporal summation and some spatial summation. So here's our little voltmeter. We're measuring the inside of the cell versus the outside of the neuron. Here, what the membrane potential is, or the resting membrane potential. And here's another neuron, a little bit of ways away here. It's sending a signal down its axon to its synaptic vesicle there. You know, it's maybe dumping a little bit of neurotransmitter in there. A few ligand-gated sodium channels open. Some sodium starts to go in at that little region. Even though it's only happening at that region, it affects the entire resting membrane potential for that cell. Okay, so a few extra positive sodium ions came in at that local region, that local area, but it does affect the entire resting membrane potential, but only a small amount. So you see this little, this was minus 70 millivolts. That one little action potential came down from another neuron. Bloop. You get a little rise, a little positive charges going in. It becomes a little less negative. So maybe that's minus now 65 millivolts that got to. And then another signal doesn't come. You know, this happened to be that one little signal came down. And then at that point, boom, nothing happens to that cell. You didn't change the resting membrane potential enough to reach threshold yet, to cause voltage-gated sodium channels in the axon hillock to finally say, and open up to allow this propagation, which we'll get to in a second. Here would be spatial summation. So here's a bunch of axons from different neurons. So there's other neurons that came down, and it could even be multiple axon terminals from a single neuron. It doesn't matter, but just they're all arriving at about the same time on that same neuron. Spatially, they're close to each other. So here's a bunch of impulses coming in all at the same time, and they're each causing some ligand-gated channels, in this case sodium channels, to allow some positive charges to come in, right? So in this case, you'll notice that because you had multiple ones all arrive at the same time, they add up. They added up. All those extra positive charges add up, so we've got a bigger spike here. So maybe we're at minus 60, 62, or not quite, or minus 60, minus 54, or but well, not minus 54, minus 56 millivolts. We haven't quite got to the critical threshold value, which is actually minus 55 millivolts we have to get to. And then no other signal comes, and it just goes back to resting. Once that signal's gone, the rest of the sodium-potassium pumps just crank it out and just gets back to resting here. This is called spatial summation. This is where we get a little summative action here. That in this case, we didn't have enough yet to get to threshold, but you could get to threshold if enough came at the same time onto the same neuron, enough little local potentials. These are a bunch of local potentials arriving on the same neuron. Another method is called temporal summation. Usually it's a combination of both temporal and spatial summation. So here's an axon that's generating multiple action potentials rapidly in sequence. So it keeps dumping more neurotransmitter to keep opening more sodium channels here, ligand-gated sodium channels, so more sodium gets in. So you see this little almost like stepwise progression where there's not enough time for the sodium-potassium pump to reset and pump them out, and they keep coming in with each successive little impulse or each next action potential that are quickly they're summing on each other to allow more sodium in. But again, we haven't reached threshold yet. We didn't quite get to minus 55 millivolts, so we didn't actually get this neuron to create an action potential. But this is the process used to get to that minus 55 millivolts. It's usually a combination of both temporal and spatial summation where enough local potentials land on a neuron and open enough sodium channels that they get to a threshold value.
That was kind of my question. I was just wondering if there's a purpose to all the little signals that like don't add up to the big signal, like if that doesn't themselves have value, or if it's just like a trio.
It can, because there's things where you can actually have, and we'll talk about, there's like presynaptic facilitation where if you have some local potentials that keep this guy a little bit closer to threshold, and then another signal comes down and gets it to threshold even more easily, more quickly. So you can set up circuitry in your system that's a little more sensitive to stimulation, because if you have these little local potentials happening, you can try to keep your resting membrane potential a little closer to threshold so that it doesn't take as strong a stimulus to get it to threshold and cause a stimulus. And so that's some of the mechanism that happens with what's called hypersensitivity. When somebody becomes overly sensitive, it can be a negative adaptation, like with pain and chronic pain, where you get some hypersensitivity and you get like this baseline resting increase and the membrane potential gets closer to that threshold. So any little stimulus, when you barely blow on their skin, they're like feel pain because you're already close to threshold. Or it can be something where you actually want to get more sensitive, so you're more hyper aware of something, or you're strengthening circuitry, like if you're doing a movement, you're learning a new movement, and you're trying to get strengthening between neurons. You might get them so they're doing this so they're staying closer to resting. So if you activate that pathway, it can activate more easily and give you a strong response and those kind of things. There's also the opposite of this. There's actually presynaptic inhibition. So there's other times we want to quiet neurons down, so you can actually open up chloride channels to get some negative charges, coming in and make it even more negative on the inside, so it's even harder to get. So that's another process to actually downregulate. Remember, everything in our bodies, all of our organs, including the nervous system, they're like set in the middle, and you can either turn them up or turn them down. And so these are all methods of turning up or turning down the sensitivity of your nervous system, is this either facilitation or inhibition of any organ system. It's usually kind of got a set point that's somewhere in the middle that you can go up or down in activity from that. So right now we're talking about increasing activity or causing things to happen that are excitable, but we'll talk eventually about inhibition, which we get to synaptic transmission. We'll talk about some of those inhibitory connections. All right. Let's take another quick break here before we just run through the action potential. All right, action potential now. We're finally getting, after 2 hours, to the actual action potential itself. So we just go a little deeper in this neuroscience you probably had in other courses, but hopefully it makes sense. So the action potential itself, the important elements of the action potential, we're going to go through each of these individually, as we first of all have to achieve a threshold stimulus. which is achieved by accumulation of those local potentials that all add up to get you to this threshold stimulus that cause the rapid change in the voltage that causes the change in the voltage-gated sodium channels. You get this depolarization, which is the rising phase, where it's getting more positive on the inside. It's getting more positive on the inside, so you see it going more positive. That's the rising phase. There's actually a period of time where it gets even more positive than 0 and goes past that. That's called the overshoot, because things are rushing in so rapidly that they actually overshoot a little bit. And then eventually you repolarize and get more back, trying to reestablish that resting membrane potential. It's going to be negative, so it's got to go back down and get more negative again. And it has kind of like you had this overshoot on the top end where it gets a little more positive than you would normally expect. It gets a little more negative on the lower end because of the same thing, where you just get a little more, and we'll show you this where this happens here. Is the hyperpolarization briefly, and then during that hyperpolarization period, you have what's called kind of this period of time. It's called absolute refractory, where you can't stimulate the neuron again. You can't re-stimulate the neuron. It stays quiet. You can't. So that's important. You can't re-stimulate those voltage-gated sodium channels again. That's why you could only propagate in one direction. So because there's a temporary period of time where once you've done the stimulation of that area of the nerve, that it goes quiet. So you can't. It has to keep going this direction, because the only voltage-gated sodium channels that can be stimulated are going to be downstream from it. Does that make sense? And so there's that absolute refractory period is really important to keep. So you can't spread a action potential both directions. It can only go unidirectionally, and that's how you can send signals in a specific direction, is because you've got that absolute refractory period that temporarily blocks the signal from going the opposite direction. And then you have a relative refractory period where you could stimulate the neuron again, or you could stimulate those voltage-gated sodium channels, but it's going to be a little harder to stimulate them. It's going to take a stronger local potential response. You're going to have to exceed even more than minus 55 millivolts to get them to respond. It's called the relative refractory period. So let's talk about threshold first. So here's the action potential. This is 0. This is down minus 65, 70 millivolts. A neuron gets stimulated by some other neurons. Okay, so these other local potentials, these other synaptic, presynaptic terminals, terminal boutons, are dumping some neurotransmitter nearby, or you're pushing on a receptor that stimulates some modality-gated, sodium channels, and you get a local potential, or several local potentials that either through spatial summation or temporal summation or a combination of those finally add up to enough sodium ions get in that you get to at least minus 55 millivolts. Okay? That will then initiate the action potential, which that causes the voltage-gated sodium channels that there's a high concentration of them in that axon hillock at that initial segment. That's called the initial segment, right at the beginning of the axon. All of a sudden, all those voltage-gated sodium channels say, minus 55 millivolts, boom, let's all open, and they all let the sodium in right at that axon hillock, right? Okay? And so that's going to get then this action potential started, okay? So now all those sodium start rushing in to make it more positive on the inside, because they're moving really fast and strong, because there's both a concentration gradient, right? Because sodium's much more higher concentration on the outside than on the inside. They're going, man, we want to go with our concentration gradient, but there's also a really strong electrical gradient because it's more negative on the inside, so they're getting pulled electrically and chemically, just being injected into this, into the inside of the neuron. And they actually they come in so fast and rapid that some of them actually kind of pile on as they're piling through there, even though they shouldn't go, they're actually exceeding that chemical and electrical gradient a little bit. So that's where you get the overshoot, we call it, where you actually go beyond 0. They're coming in so fast with momentum, they kind of then they keep you shooting even above establishing that electrical, neutral 0 millivolt difference between the two. So that's kind of the overshoot here. So here's the depolarization rising phase. So this is showing you what's happening here as it's depolarizing very rapidly. The sodium ions rush through open sodium gates, the voltage gated sodium gates, due to that large concentration gradient and electrical gradient. Inside of the cell becomes more positive. If threshold voltage, then the voltage gated sodium channels open. And then potassium channels, though, remain the same during this phase. So potassium channels are just kind of sitting there. You have leak channels, but there's also gated potassium channels. The gated potassium channels are remaining closed at this point. Okay, so we still have those leak potassium channels, but remember the sodium potassium pump's still working this whole time, throughout this time. But so right now, just worry about sodium. It's rushing through. Okay, and then, like I mentioned, the sodium rushes in so quickly that it actually overshoots, and we get up to typically like a positive 35 millivolts on the inside of the cell from all that sodium rushing in. Okay? Now, repolarization is the falling phase where we start to reset the cell. Okay? And so the way that works is that the voltage gated sodium channels, those voltage ones have a timing mechanism. When you reach that threshold value, they open for a few milliseconds. Then they just start to close automatically. Okay? So they're closing. So sodium can no longer pass between this. But now, in addition to the leaky potassium channels, you also have a bunch of voltage gated potassium channels that have been delayed. There's a delay mechanism on these voltage gated, so they open a few milliseconds after the sodium channels are open and then start to close. Okay, and so then there's a little delay, and now we get this voltage gated potassium channels starting to open. Okay, and as the sodium channels are closing. Okay, so now when those open, now we also have because you had all those sodium ions rush in, it's more positive on the inside of the cell. Plus you have more potassium on the inside and the outside. So now potassium wants to move out of the cell very rapidly because it's got a strong concentration gradient and electrical gradient pulling and pushing it out of the cell. Okay, so we've got the opposite thing happening now. So potassium's rushing out and pulling and causing this rapid drop in repolarization. So you're losing positive charges. Remember, in the background, as this is happening, the sodium potassium pump's also working. So the sodium potassium pump's in the background also pumping out sodium, but pumping some potassium in, but it doesn't do anything to counteract when all those voltage gated potassium channels open, how fast those will rush out. But just remember, you always have that sodium potassium pump in the background. But the most of this is driven by this opening of these voltage gated potassium channels above and beyond the leak channels. Okay, so then it rapidly drops down, and it happens again so fast that you actually lose a few more potassium molecules because of that big concentration electrical gradients that you actually go past very briefly below the resting membrane potential. And that's that then that's part of the absolute refractory period when we get there. That's the hyperpolarization. So here's the hyperpolarization right here. And then then after a certain few milliseconds again, now everything's closed again. Okay? And so when those are closed, there's a period of time where you cannot reopen those. And that keeps those from being re-stimulated, and it keeps you from, and it makes that you have to propagate the signal down the line, because it starts at that initial segment. You open those voltage-gated sodium channels, that initial segment, they open, causes depolarization, which causes then depolarization along down the line, and we'll show this jumping of this charge, stimulus down those nodes of Rabier. But then temporarily, that initial segment is not able to be stimulated again, so it can't go back this way. It has to go that way from the initial segment. Okay. And then after another few milliseconds, you eventually get back to your resting membrane potential. So there's a period of time, I think I've got a picture of an absolute refractory period where you can't stimulate these. All of these are at a point where they can't be open, but then there's a period of time where some of them are starting to reset, where they can be open, but not all of them have reset yet, where you could stimulate them again, but it takes a stronger stimulus than even minus 50, minus 55. You might have to do minus 60, or minus, not minus, it's kind of more positive, should be minus 50 millivolts or whatever to get it to stimulate. This is just showing you what's happening, during certain phases here. So this yellow thing is just showing, this is when you're getting some to open to these local potentials, right? The local potentials are adding up. Then you hit threshold right here. So some sodium channels are opening because of, other neurons causing, the cell to start to depolarize. And then once you hit that minus 55, now the voltage-gated sodium channels open. That's this green phase, this rapid rising phase. This is the voltage-gated sodium channels being activated at the initial segment. And then those start to close, and at the same time you get the sodium channels close, then you get the potassium channels opening, the voltage-gated potassium channels, which are even a little further delayed than the sodium channels when they start to open. And then that's when you get the falling here, because the potassium is now rushing out to take positive charges out of the inside of the cell, while sodium is also being pumped out of the cell by the sodium-potassium pump. So that does contribute to some of this falling of that, because you're trying to reestablish resting membrane potential. And then it temporarily drops just a little bit below and then recovers. Once all the channels are closed again, the voltage-gated channels are all closed, and the resting membrane and the sodium-potassium pump just reestablishes that resting membrane potential. So here's the absolute refractory period. So during the rising phase and then partway during the falling phase, either the sodium channels are open and they haven't been reactivated, and the cell's too positive. You can't re-stimulate that. You can't re-stimulate it when it's already been activated. There is a point, though, once you depolarize the cell enough, get it back down where it's starting to get negative, that you could reactivate or cause another action potential. It's going to require a stronger stimulus to do that, okay, during that relative refractory period. So it's during this whole time window here that it'd be much harder to reactivate or cause another action potential. So, because just the gated channels aren't ready and reset to allow that. And you haven't restored the concentration gradients and so forth, electrical gradients, enough to be able to allow that to happen as easily to generate another stimulus. So that's what I just talked about there. Now we're going to talk about, so once you've activated that initial segment, those voltage gated, and you have that rapid change of charge, it will then cause voltage-gated sodium channels down the whole axon to start opening in sequence. Okay, so you have voltage-gated sodium channels down your axon. That once you've opened those voltage-gated sodium channels and caused that flip-flop of charges at the initial segment, the ones nearby will recognize that and feel that charge difference, and then they will also start to change and open and cause the propagation of the action potential down the axon. And there's three things that influence how this occurs, or how fast this can occur. One is what we call axoplasmic resistance. Axoplasmic resistance is simply the resistance to charge moving kind of passively within the axon itself. So when you're getting this flip-flop of charges, and the charge will actually passively influence a certain distance away from, that'll influence, and a certain amount of charge will passively move within an axon. The axoplasmic resistance is what provides resistance to charge moving passively a certain distance within the axon, influencing the voltage-gated sodium channels a certain distance away. Okay. It's kind of like a hose. You know, water moving through a hose. Charge will actually move through the cytoplasm in that axon a little ways. The bigger the hose, the easier it is for water or the volume of water to passively move through a hose. The smaller the hose, so bigger axons, and they actually include the myelin in the diameter. So a bigger myelinated axon, the larger, it's easier for charge to passively move because there's less axoplasmic resistance to charge just moving down the line. Okay, that's RA, axoplasmic resistance. Then there's a thing called membrane resistance. Membrane resistance is the resistance or really it has to do with the ability of ions to move across a membrane. The more permeable something is, the less membrane resistance it has. The less permeable, the more resistance it has for charge to move across it. Okay, so the membrane resistance to charge moving across it. And then there's membrane capacitance, which is the ability to store charges across a membrane. Okay, so that's when you get that lining up of positive and negative charges right along the membrane there. That's storing the charges right next to each other. The closer they are, the easier it is to store those charges next to each other, which can be, we'll talk, it's kind of counterintuitive how this influences propagation of the action potential. But the two characteristics of nerves that affect the above is the diameter of the axon and the myelination of the axon. As I mentioned, the diameter of the axon, the larger the diameter of the axon, the easier it is for charge just to move passively through it because that lowers axoplasmic resistance. There's just more room for ions, for things to move in there and just let charge flow. Okay? Myelination, so myelin, this insulating stuff, it has a unique feature. It increases membrane resistance. So it actually makes it harder to store charges across the membrane because it increases resistance. It makes it so those areas can't be permeable, right? Because there's actual physical barrier, so there's no permeability there. Actually, I was going to confuse capacitance with resistance. So it resists charges moving across that area of the axon because it's an actual physical barrier. Which at first you think, why would it do that? We'll show you in a second here. The other thing it does is it decreases membrane capacitance. It makes it harder to store charges across that area because it creates a gap. And so not only can they not get through there because there's no proteins, there's no pores to get through there, but the positive and negative charges are far enough apart that they don't interact with each other. You can't store the positive and negative charges right next to each other, so they don't interact as much because you've got a distance between them that makes them influence each other less, those positive and negative charges. So let's just kind of talk about what that means. So here is a axon that has no myelin on it, right? So here's the initial segment. We get some local potentials here that cause it to depolarize enough that we eventually cause these voltage-gated sodium channels to open. Boom. We start an action potential. Sodium rushes in. And if this is a smaller diameter axon, the charge passively moves. It's harder to move charge far enough to help influence these next voltage-gated sodium channels down the line. It makes it a little harder for them to influence that because they just don't passively spread as easily or as far. The other thing that happens is that because you have no myelin on here, myelin that kind of surrounds this, is that you have actual storage of charges right next to each other. On each side of the membrane, you have these positive and negative charges lined up. And what that means is you actually have to then go and get this flip-flopping of charges down the line at every single one of these. I'm trying to think of, it's almost like, analogy would be like sticky Velcro. If you, the positive and negative charges are like sticky Velcro, and the harder you push on that Velcro, the harder it is to quickly like rip Velcro apart. That's what's happening here. It's actually because you have so many charges stored there, you have to. Actually slows down how fast you can get that flip-flopping of charges, because they're kind of bumping into each other, getting in the way of each other as you're trying to get this charge to passively spread down the line, and you have to get this flip-flop all the way down the line. That's the problem with an unmyelinated and small axon diameter that makes it harder to spread. We do have those. We have small unmyelinated, and that's why they propagate charges slowly. They're kind of narrow, so they have lots of exoplasmic resistance. They have a lot of they have less membrane resistance. So they actually, there's more permeability and there's more ability of those charges to move across. But that means that you have to do that a lot, and they kind of get in the way of each other. And then you actually can store more charge across it, which again can make it a little slower, and we'll show you why here in a second. So here is what myelin does. So myelin here's our initial segment here. We get our action potential, and we get positive charges moving in. The myelin actually, for some unknown reason, but it also contributes to the diameter. So when you talk about a diameter of an axon, you include the myelin. So the myelin actually makes it function like a bigger neuron, but you have the diameter of the axon here. If this is a larger axon with myelin on it, this positive charge will spread very easily and move passively a certain distance. And it just so happens our bodies and our nerves know what that distance is. And it's the distance between the two nodes of Ranvier. So this is a node of Ranvier, is these little open, these are exposed areas of membrane on the axon. And so what happens is, boom, you get the positive charges coming in from sodium. That positive charge can freely move a certain distance in an axon and affect the next voltage-gated sodium channels that are exposed here, that where all you need to do is store a little bit of charge at each of these, and it'll jump. Essentially feel like it's jumping from one to the other. Versus, if you have all these, positive and negative charges lined up all the way along, you have to get each one of these to kind of flip-flop, flip-flop, flip-flop all the way down, and it doesn't allow you to take advantage of that passive spreading of charge. And you're storing all this charge along there, and they get in the way of each other, of those ions and everything. So where the myelin is, there's no protein channels, there's no ions in the way, and that allows the sodium ions and that charge to spread passively very quickly and unencumbered to the next node of Ranvier. And then all you need to get is just relatively few sodium channels at that next node to flip-flop to passively spread to the next one. And so that's the difference between, like, if you took like two pieces of Velcro, and without myelin, you crunched them together and you really pushed them together, like they're really sticky and a lot of charges are stuck together. And as you pull, you have to get those charges to break apart or flip-flop, and it's hard to pull it apart. It takes longer. Versus if you take that same two pieces of Velcro and you just tack it down here, tack it here, tack it here, tack it here, and then you go, and it just rips apart a lot easier. That's the same process that this charge jumps down these nodes of Ranvier. I don't know if that helps or not. That, or those pants you flip up when you had the buttons down the side or whatever. It just makes it easier than having the Velcro, because it's like the nodes of Ranvier. You just kind of rip it off because you can passively move that down the line. Does that sort of make sense now? So it is kind of counterintuitive with these things that you don't want to have, you want to have less exoplasmic resistance. You want to have more membrane resistance in certain areas to keep, so those charges can't move across that area. And you actually want to have, less membrane capacitance in areas, but only for certain. You got to have it in certain areas. You can only get away with this. You can't myelinate the whole thing without any nodes of Ranvier, because you can only passively move that charge so far. So it's ideally set up so it'll jump from one to the other based on the ability of our natural ability to passively move those. Now talking about some general interactions between neurons when we're neurons are talking to each other or sending signals to each other. Um, the diversity as well as the specificity of the nervous system can be attributed to these connections between our neurons. So there's different ways our neurons interact, and one thing is we can do is with a neuron we can focus a signal. Focus a signal or pathway is through the thing called convergence, and the convergence would be where multiple neurons are focusing on a single neuron. Okay, you're focusing a signal for a certain specific activity. If I'm going to move just a little tiny muscle in my finger, it starts out with a lot of neurons up in my cerebral cortex, and then eventually that signal gets focused down to just moving a select few muscle fibers for a fine control or something like that. That could be an example of kind of convergence that happening throughout the nervous system. And this happens at a broad level like that, but also can happen at very local circuitry levels within the spinal cord, with reflexes, within little regions of your brain, those kind of things. You can have convergence, where you've got multiple neurons focusing a signal to enhance the activity of a single neuron or a few neurons. And then the opposite, you can have divergence, where you have a single neuron influencing more than one neuron when you want to diverge a signal and expand a signal to multiple neurons. So all these are ways of regulating activity in the nervous system, either upregulating, downregulating, making it more complex, making it more narrowed. These are just ways of our neurons interact with each other to influence activity. I already mentioned these for clinical considerations, the Guillain-Barré and the multiple sclerosis. Also, just understand when you're looking at a patient's chart for the hypokalemia, when you're looking at electrolyte imbalances, the one you want to be really most concerned about is potassium. Anybody want to take a guess why, based on what we just talked about? Why would potassium levels in somebody's blood be more of an issue than sodium level as far as clinically for you, based on what we know about resting membrane permeability? Yeah, because of the leak channels. So especially if you have rapid changes where your body doesn't have a time to adjust, if you get a rapid change in potassium in your blood and your extracellular area, that can influence your nervous system by changing the ability of your nervous system's resting membrane potential. Either it could be closer to hyperpolarized, where it could be hyperpolarized, or you've got too little potassium and it becomes hyperpolarized, so it's harder to actually get an action potential, which could be bad if you're trying to activate neurons that are important for maintaining breathing and your consciousness and those kind of things. Or it could be the other thing where you get closer to resting membrane potential, that threshold value where you might get overactivity, where you're getting muscular spasms, you're getting cardiac arrhythmias because you're getting closer to threshold. So just understand that too little or too high calcium that happens pretty rapidly can be an emergency kind of event that can influence, can cause muscle spasms, can cause arrhythmias, it can cause loss of consciousness. Just be if you're going in treating a patient and looking at their electrolytes and you see some, change in potassium. Hey nurse, I just noticed their potassium's all out of whack. Are they getting potassium? Are you going to try, or are you waiting to get rid of some potassium for them? Should I see them right now? Those are the things where sodium doesn't matter that much, because sodium, you can have high sodium levels or low sodium. Well, low sodium can eventually, but it's not as problematic. It's not as immediate. It's not as profound. The changes when you got a little bit more leeway with sodium differences in your system. So. Unless the sodium levels get really low, then that can be a problem. The hyponatremia, but that's hard. That's a harder condition to get or achieve. So, are we done yet? Almost after this picture. This is just showing you some of the. This is an MRI, just showing you some of the lesions here within somebody who's got multiple sclerosis. And you'll notice this actually, so the lateral ventricles are just kind of sitting right in this region here. So you can see these lesions tend to be, in this case, located near some of the lateral ventricles. And really near the lateral ventricles is where a lot of your white matter tracts run, so your motor and sensory tracts run down through there. So that's why it's very common to see hemiparesis or paresis. This person could have paresis on both sides or sensory problems on both sides because they have lesions bilaterally. Um, and it can be very sporadic because some of the lesions can be so localized that they might have, like, they're numb right here, you know, and that could just seem really interesting that they're numb right here, which would seem more like maybe a peripheral problem, but then they have spasticity in their legs also because of the lesion back here, you know, that kind of thing. So that's why MS is really interesting, but also, you know, frustrating, sometimes confusing because the symptoms can be so varied with the patients. So. Okay. Just some things, clinical considerations for drugs and toxins. Some of those are used to inhibit sodium channels. So a lot of your local anesthetics, such as lidocaine, block sodium channels. So you can't, when they stick that needle in, once it's numbed up, you know, you can't reactivate those. You block those sodium channels, so you can't send a painful, you know, action potential signal impulse from one of your pain receptors, you know, because you block their sodium channels. The puffer fish toxin, which is tetrodotoxin, is a powerful inhibitor of sodium channels, which can cause you to stop breathing if you inhibit the neurons that are important for maintaining breathing, or stop your heart. So just a couple things. And I'm sure Dr. Renner will be talking about other drugs and things that can influence sodium channels and so forth, and potassium. Any questions before we turn you loose? A little early. So that's good. A lot of times this lecture I end up having to extend it a little bit to the next session. You guys are fast learners, so I don't have to slow down for you.